Hi guys, so today we're going to do a short review on carbohydrates. We're going to focus on the main elements between monosaccharides, disaccharides, and polysaccharides. So if you guys remember carbohydrates, right? It's carbohydrates, we have carbon, hydrogen, oxygen, and the little N right there, right? So even though the term carbohydrates, it sounds like it's molecules of carbon connected with water molecules, but it's a little bit misleading because they're actually not. And the smallest, actually we're going to put like an N right there because the smallest carbohydrate is going to be a triose, right? So in order to start like to make a difference, we're going to be covering monosaccharides. I'm going to put just like very simple, right? Disaccharides and polysaccharides. All right, so we're going to get started with monosaccharides first. In order to make a difference, these are going to be your simple sugars. So I always try to connect the molecules associated with food and a little bit of nutrition and diet. So you can start kind of like understanding the role that the carbohydrates have in our diet, right? So we're going to have the monosaccharides. Now, in order to start with the monosaccharides, there are going to be some basic difference that we can make a difference between all of those monosaccharides. So we're going to have 1, 2, 3, and 4 differences, okay, that we mentioned in class. And number 1 is going to be the presence or that location of that carbonyl group, right? So the carbonyl group is just going to be very simple. Carbonyl group is also known as a ketone. So you have probably heard that term before in diets referred to as ketones. Well, ketones is nothing but actually a carbonyl group. So this is a carbonyl group. Carbonyl group. And let's remember also to make a difference between carbonyl group and carboxyl groups, because in amino acids, we have actually the term carboxyl groups, which are elements that are like part of your basic amino acid structure. So in carbohydrates, don't get confused, we have the N right there. So it's going to be a carbonyl group. And that carbonyl group is just going to be a carbon with a double bond connected with the atom of oxygen. And that's going to be, yes, a ketone right there, right? So all of these simple or monosaccharides, they have attached that carbon with a double bond to an oxygen. It's actually considered a ketone, right? So to state the example, the first number 1, the location of that carbonyl group is gonna make it's gonna matter, and it's gonna make like a very different type of monosaccharide. For example, we have aldose and ketose, and if you notice that the molecular formula is actually the same. It's pretty much identical in terms of the amount of carbons that it has. It has 1, 2, and 3 right here. 1 carbon, 2 carbon, and 3 carbons. So it's going to be a triose, right? Because it's a 3-carbon molecule. And that's actually going to be the smallest type of monosaccharide or sugar that we can find in carbohydrates. Now, if you notice, the only difference, if you notice the presence of the hydrogens, the hydroxyl group, the hydrogen, the oxygen, the only difference between the aldose and the ketose is actually going to be the location where that carbonyl group is going to be located. And on the aldose is going to be located right here on the carbon number 1. So that's going to be your aldose. And for the ketose, which is a ketone type of sugar, right, it's going to be located on your carbon number 2. So that's it. That's very simple. Just making the difference is going to give us either an aldose or a ketose or a ketone type of sugar, right? Just the location of that carbonyl group on the carbon number 2. And that's it. So that's going to be a 3-carbon molecule, a 3-carbon monosaccharide, very simple. And the only difference in terms of structure is just going to be the location of that carbonyl group. That we're going to flip the components from carbon number 1 to the carbon number 2. All right, so that's number 1. So in order for carbohydrates to be considered a carbohydrate, going a little bit back to that, we have to consider some important elements. Number one is going to be that they have a CO, a carbonyl group. So they have the presence of a carbonyl group, right? Another thing, and we're going to jump right a little bit into that, is going to be the presence of hydroxyl groups. So they have a lot of those hydroxyl groups. You can see right here we have 1, 2 in that aldose, and we have another hydroxyl group and another hydroxyl group right there located. So pretty much it has the same structure, right? The same components. So it's going to be the presence of those hydroxyl groups, and it's going to be also a lot of carbon to hydrogen bonds, right? So we have a lot of carbon to hydrogen, carbon to hydrogen, carbon to hydrogen, carbon to hydrogen. So you're going to see that in all of these molecules, those will be kind of, we can consider the 3 main components of the carbohydrates, right? The presence of the carbonyl group, just to go again, the presence of those hydroxyl groups, and the presence of a lot of these carbon to hydrogen bonds as well. Okay, so also, so we already have one difference between, to distinguish one monosaccharide between another monosaccharide. The second difference, if you guys remember, is going to be the number of carbons present in that molecule, right? So we have the total number of carbons right there present. The number of carbons. So we can go for the smallest type of monosaccharide, which is going to be your trioses. Triose is going to be the presence of 3 carbons, right? Like aldose and ketose again. Another one is going to be, for example, your ribose. Your ribose is going to be a total of 5 carbons in that molecule. And then we have the typical, that's your standard typical carbohydrate monosaccharide molecule, and that's going to be your exose molecule, exose. And we're going to put 6, right, in that case. So we have a triose, we have the triose right there, like the example of the aldose and the ketose. We have the ribose. If you guys remember the ribose, riboses are very important to help, kind of like to be part of the building blocks of bigger molecules, like other macromolecules, like nucleic acids. So if you guys remember, we have the ribose or the deoxyribose. And I put ribose there. It actually should be a pentose. I was thinking already over the ribose, right? But this actually, the proper name should be pentose, right? I'm sorry, guys. So that will be a pentose right there. So we have the pentose. It's going to be a total of 5 carbons, right? Remember that we always start counting here. 1, 2, 3, 4, 5. And it can be a ribose, like in RNA, and we can be a deoxyribose, like in the presence of DNA, right? And then we have the typical 6-carbon molecule, right? Which is going to be your 6 carbons. So 1, 2, 3, 4, 5, and that will be 6, right? Pretty much like that. So that's going to be your exose, a total of 6. And glucose will always be your typical, like glucose, making the representation for carbohydrates, right? So we're going to have that. And then we're going to move to number 3. So for that, we're going to have the spatial orientation of some components, in this case of the hydroxyl group. Right. So where that hydroxyl group is located, it's going to matter. It's going to tell us the type of sugar that it is. Kind of like in the example of the aldose and the ketose, it's going to be, of course, something very similar to the glucose, for example. So let's go ahead and take a look at the molecule of glucose. So this is going to be a linear representation of a molecule of glucose. Let's remember that glucose is composed of 6 carbons, right? We have it right here. So it's going to be carbon number 1, carbon number 3, carbon number 3, carbon number 4, carbon number 5, and your carbon number 6, right? So we have the location of the carbonyl group located right here on our carbon number 1. It doesn't want to go ahead and, there we go. Well, on carbon number 1, we have the carbonyl group. We have all of our carbon to hydrogen bonds as well being connected there. And if you notice right here on the carbon number 4, we have the hydroxyl group, like with the spatial orientation kind of like facing upward, right? If I were to make a flip and switch that hydroxyl group, like facing down, looking down, instead of that position, right? Let's just say that I'm going to switch it. In glucose, it should be facing up, right? If I go ahead and flip these two, the hydroxyl group with just the hydrogen right there, it will give us a completely different molecule. It will give us galactose. And just like that, it gave us a completely different type of sugar, just by making a small, like tiny arrangement in that molecule, right? So we have glucose, and then we're going to have galactose in that carbon number 4 with the hydroxyl group if it's facing downward, right? So we have that. So we have that another difference. And final is going to be the linear organization, linear versus a ring structure. So a ring structure, I'm going to put it like that. So let's just say that I go ahead and take that glucose molecule. Let's forget about the galactose. I'm going to focus on the glucose molecule. So I go ahead and pretty much draw my, I'm going to put it like very basic, okay, like an exos type of sugar. Okay, so if I go ahead and attach this carbon number 1, right, and usually, for example, when you put these molecules of these carbohydrates in water, they're mostly going to take a ring structure. So if I go ahead and attach them and connect them together in a circular form, right, so I'm going to have the carbon number 1, 2, 3, 4, 5. Technically, we have that 6 right there, but always it's going to be over here, like facing all over there. So if I go ahead and put that hydroxyl group facing down, and we have hydrogen bond right there. So if I put that, how I arrange, I'm so sorry, those molecules connected, these ones over here. I was thinking about the hydroxyl group of the carbon number 4. But if I go ahead and an arrangement, and when I'm connecting them, I end up with having this hydroxyl group in the bottom. I'm going to have an alpha type of glucose. So I'm going to have a glucose alpha right there. So it's going to be an alpha glucose right there. If I end up with a hydroxyl group on a different direction, right, facing up, so I'm going to put kind of like 2. If it's facing down, it's going to be alpha glucose to have like a visual representation. I always need my visuals. But if I go ahead and switch it and I end up with a hydroxyl group on the top instead of the hydroxyl group, then it's going to be a beta type of glucose. I'm going to put glucose, and I'm just going to put it facing up, right? So that's going to be a beta type of glucose, right? So that also is going to matter when I start connecting maybe these monomers or these saccharides with other molecules and join them together at that structure, right? So if I go and connect that number 1, right, with another monosaccharide, another molecule of glucose right here, right? And I connect them over here. If you notice, this is going to be the carbon 1, the 2, the 3, and the 4, right? So if I go ahead and use a glucose alpha right here, this is going to be an alpha, and I'll just go ahead and flip it. Oh no, it's going to be another alpha glucose right here. Okay, I'm going to end up, I'm going to delete that hydroxyl group. I'm going to connect them like that. So I'm going to be able to form a 1,4 glycosidic link. Glycosidic link or bond, however you want to call it. So you see the 1,4 or the 1,6 or the etc. Those numbers represent how those two different monosaccharides are being joined together in that location of that hydroxyl group, right? So in this case, the first molecule is going to be the 1, and right here is the carbon number 4. So pretty much that indicates the position of that glycosidic link, right? So again, we have these 4 ways to make a difference between one monosaccharide and the other type of monosaccharide. So we have that, right? So I'm going to put as examples right here. Let's use the green one. So I'm going to put here the glucose. It looks like the yellow. Glucose. galactose, and fructose, right? So we have those three different types of monosaccharides. So when I'm talking about disaccharides, that means two, right? We're pretty much going to be making connections, these glycosidic links, connecting two different types of monosaccharides together. So if I put glucose and connect it with another molecule of glucose, so glucose, excuse me, polysaccharide, I'm going to scoot it over here, and maybe right now I will make your own space. So if I put glucose and connect it with a glycosidic link to another molecule of glucose, right? That's going to be, we're going to end up with a maltose, right? So it's going to be a maltose. So maltose is a type of disaccharide. It's a type of reducing sugar. We call them reducing sugars because when you absorb them, they pretty much have to break away that glycosidic link in order to release the two molecules of glucose, and those two molecules of glucose be able to be taken in, absorbed at your gut, right, by your receptors, and be able to work their way into the cells. Now, for galactose, for example, we're going to connect a molecule of glucose, glycosidic link, with a molecule of galactose, right? So we have galactose right there. Again, we have that glycosidic link, and in that case, we have lactose, right? Lactose. So lactose is a type of disaccharide. So all that is found in your milk is actually a type of sugar, right? It's a type of reducing sugar. So when you're drinking milk, right, your milk has to be able, by your enzymes that we have in our intestine, like lactase, right, has to be able to have the ability to break away that glycosidic link and again increase the presence of glucose and increase the presence of galactose so they can be absorbed in your intestine. We're going to have an activity related to that in the lab, so we're going to be working with the reaction of galactose and be able to measure the concentration of glucose and to see how that lactase is actually working in that solution, right? So remember that. Now, finally, we have fructose. Fructose is pretty cool. So we're going to have sucrose. So for example, for glucose, I use the green one. So we're going to have glucose. And then if we join it together with a molecule of fructose, which is a type of ketone again, right? So the sugar that is found in your orange juice, right? If you think directly like the juice from orange, right? It's a type of fructose, so it's a type of ketone. So that's why it's like it will be easier quickly to absorb. Let's just say that you're feeling a little bit of hypoglycemic with your low sugar to take some juice because your fructose is going to be able to be absorbed like very fast, like the same as glucose, right? So instead of thinking, for example, like table sugar. So we have here table sugar, right? That's your table sugar. That's sucrose. Let me put the right name. So sucrose. So we're going to have sucrose. That's your table sugar. So if you think about it, if I'm here taking table sugar and I'm here taking orange juice, right, and I'm feeling a little bit hypoglycemic, which one do you think will act faster for faster absorption? So it would technically be a fructose, right, because that one is already reduced, so it will be absorbed faster. And sucrose will have to be working their way or wait for an enzyme to be able to break away that bond in order for the fructose and glucose to make their way into your bloodstream, right? So just to quickly see the differences between those sugars and why all of these links matter, right? So we already have our monosaccharides, we have our disaccharides. Let's go ahead and move on to the polysaccharides. So for that, I'm going to quickly delete this one right here, so we can make our way to talk about polysaccharides. And I'm going to delete this, okay? I'm considering you guys already know this by heart. All right, so let me delete that. We're good to go. Let's use the green one. It looks like that one. It's easier. Okay, so let's go ahead and talk about the polys. Now the polys are a little bit more, they're not complicated. Polysaccharides. I'm going to put the polys just like that, right? They're just like connected. They're a little bit more complex molecules. They pretty much have the same, obviously they have the same components of all carbohydrates, the carbonyl group, the process of hydroxyl, and a lot of these carbon to hydrogen bonds, right? That all of these, we know, they tend to make them like polarized molecules because of the location of those electrons, right? So they're very easy to break away when your body needs energy in comparison to fats. But okay, polysaccharide. Let's talk about polysaccharides. So number one, we have starch. We have starch, and one example of starch is going to be amylopectin. Amylopectin, right? And I'm going to put right here glycogen in the bottom because they're actually very similar. I'm going to put starch and then I'm going to put a glycogen right there. So starch is going to be a component that you are able to find, for example, in potatoes, in food, right? There are polysaccharides. We know that potatoes give a lot of energy, right? So they have a high component of, they're mostly composed of starch. And what basically starch is just both starch and glycogen, they're composed of a lot of these exose molecules, molecules of glucose joined together by branches. So I'm just going to go ahead and put like the branches right here, make my beautiful drawings for you guys that I always do. And I need to work on those, right? So I'm just going to put a branch right there, right? And the fun thing about it is that when you're making those branches, oh, these markers are not helping me today. Let's go ahead and do that. Yep. Nope, not help. Let's see, let's try this one. Okay, so I'm just going to go like that. Sorry. So the branches is just going to be that they're connected by these exose molecules, right? A lot of these glycosidic bonds. Ay. There we go. You know, guys, I always struggle with making my geometrical shapes in class, right? So the beauty of it, right, and the not so complexity, but the beauty of these molecules is that they're just composed of branches and branches of exose molecules. And they are what we call our complex carbohydrates, right? Polysaccharides, they're like our complex carbs. So if you think about your diet, right? Complex carbohydrates, complex carbs in your diet. So we have our simple sugars, right? Your quick sugars, those sugars that are just you're able to find like very quickly in your Gatorades, in your soda, in your candy, in your fruit juice, in your milk, right? That are able to deliver sugar really fast. But on the other side, we have the complex carbs. And you think in terms of diet, we have your rice, we have your potatoes, we have your bread, you have your pasta, right? So those type of foods are very good to help you reserve in your body a lot of energy. So the only difference between starch and glycogen is going to be that starch is found in some food, like in your cases of your potatoes, right, or your rice, right? So they're going to be just branches of these exose molecules right here, glucose. So glucose, this is definitely not helping. Glucose, 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 glucose. Now, in the case of, for example, starch, we have these branches like one, like every 30. There's like a branch being formed of glucose molecules. Now we have glycogen. All of this food that you're eating from potatoes, right, it's going to be reserved. It's going to be like, reserved in your body. It's going to be served as a storage for energy when your body actually needs it. If you notice, like those marathon runners, right, they're always like carb loading days before a race because they gotta be able to store a lot of these carbohydrates in their muscles as well as the liver. So glycogen is reserved in your muscles. Oh, this is not helping. Let me see the pink one. I just don't like this one because this one tends to, oh, today is working. Thank you. All right, so glycogen. So glycogen is going to be in your muscles and in your liver. So like we were talking, right? For example, these marathon runners, they usually have to run miles and miles in like very long distance workouts, like a race of 26 miles. But for that, they're required to have like a lot of these complex carbs being reserved as a type of glycogen in their muscles or liver, right? Because when they're pretty much burning all of this amount of carbohydrates in their body or energy, they burn twice as fast as 2 times of carbohydrates as like somebody will do in a lower type of aerobic exercise, right? So they have to make sure that your muscles have a lot of these molecules being stored in their muscles. So they even gain a little weight, a couple of pounds every time, because they need to make sure days before that they reserve all of these molecules of glucose in their muscles. So that's going to be glycogen. So remember, starch is going to be found in your food, like potatoes, rice, bread, right? And glycogen is going to be found is a way that we store that energy in your muscles and in your liver. And it's very important to know that. So every time that your body actually requires energy like very fast, what your muscles are going to do, they're going to start breaking those glycosidic links, right? And releasing those molecules of glucose so they can again be absorbed, go into your bloodstream and directly into your cells so they can be broken and be able to deliver that ATP really fast. And we're going to be talking about this glycolysis process in the next unit. So be ready for that. Okay, so we have starch, we have glycogen, we have cellulose. So cellulose is a type of glucose that I actually find, but you find it in the structure of your plant cells. So if you notice, you're going to notice in your microscope when we're looking at the cells of plants in the lab, you're going to see that the structure, they're very organized. There's like a very high orientation and structure of the cells. And that's pretty much happens because the cells, they're flipped every other. So let's just say that we have the molecule of glucose right here, and I'm going to flip the other molecule facing the other way. It's the same molecule, okay? I'm just trying to make it kind of like, I'm kind of like flipping it, kind of the other way. So it's going to be flipped every other way, these molecules. And by flipping all of these molecules, we are able to have this organization of that glucose molecules. Otherwise, they end up being kind of like a ladder in the case, like in a starch of like glycogen. So by flipping them, and so it's so amazing to see how evolution led to these changes in all of the biodiversity of life, right, including plants and so on. But anyway, just by flipping that molecule, we are able to see the changes in the structure of cellulose. So you will find that in your plants, right? Then in number 4, we're going to have chitin, right? So chitin is a type of polysaccharide, but in the case of chitin, the only difference is going to be they're also arranged and flipped every other. The only difference of this one is that these guys are going to have attached glucosamine. What's the full name? I know it's glucosamine. I know it's glucosamine. It's a glucosamine molecule right there. I think it's NAGA. I think so. NAG. So I know they're going to have that attached of that glucosamine structure attached as part of that glucose molecule, and that's going to make it to have that, it serves as an exoskeleton in insects, right? So now you know that insects, they're covered in sugar. So I've never tried an insect, and I don't want to, but if you do, probably I'm assuming they should taste sweet. I hope, but we know that, right? Okay, chitin. So chitin, remember, is the exoskeleton of insects, and they're made of these complex polysaccharides. They're also flipped every other. They just have that glucosamine molecule attached to it. And lastly, I forget if it's just 5 of them. we're going to have peptidoglycans. Peptidoglycans. So like the name implies, right? Peptidos, so peptides. It's going to be peptides, right? And glycans. So we're going to have two different things. We have peptides, which is going to be components of amino acids, right? Attached to a molecule of these polysaccharides of glucose, right? So they're going to have glucose, and they're going to have an amino acid residue, like right there on the bottom of that molecule, right? So we have that, and let's just assume that they have that little R attached, like right there to it. It should be actually an amino acid attached to it. And these are very important because remember that in our cells, we have these peptidoglycans right here. I'm just going to put them like there. And these peptidoglycans can act as signaling to connect with other molecules. For example, based on your type of red blood cell, right? You have a type A, we have a type B, we have type O or type 0 with the lack of antigens, right? So those, for example, are based of peptidoglycans, right? That helped out with the cell recognition of each one of these red blood cells. So also, for example, when your immune system is requiring some type of assistance, that they detect an antigen or the presence of a virus, and they need to activate the whole either adaptive or innate immune system, they're going to help and be able to send some forms of communication and chemicals between all these chemokines in order to start activating the immune response. So I think that's all, guys. A quick review of key elements of carbohydrates. I'm sure there's like more things that we can discuss, but as a general overview, I think that's it for carbohydrates. Okay, if you have any questions, please let me know. Send me a message, and I hope this works for your exam, upcoming exam. All right, thank you, guys. Let me turn this off.